For most of us, if we could just act on what we already know about health, finally change those two or three basic health habits we've been meaning to change, we'd experience a dramatic improvement in overall well being. But knowing what to do and actually doing it are two very different things. We need an approach that supports true personal health transformation. And this is why I launched a new membership based health service called Adapt 180 Health. As a member, you still get answers from licensed functional medicine practitioners and even custom nutrition plans from licensed nutritionists, but it doesn't stop there. You also get your own board certified health coach, an expert in lasting behavior change. Get the support to truly transform your life and unlock more health, joy, and resilience. Visit adapt180health.com. That's adapt180health.com to learn more and download our free ebook. Hey everybody, Chris Kresser here. Welcome to another episode of Revolution Health Radio. This week I'm excited to welcome back as my guest Raja Dhir, who is the co-founder and co-CEO of Seed Health, a microbial sciences company pioneering applications of microbes to impact human and planetary health. Raja is a wealth of knowledge on all things microbiome, and today I'm going to talk to him about the relationship between the microbiome and COVID, because there's some interesting new research out on that, as well as some updates on microbiome and probiotics and IBS, the gut-brain axis, particularly with the specific focus on depression, and the gut-heart axis. So let's dive in. It's a pleasure to have you back on the show.
Great to be here, Chris.
So I want to dive into some recent research that was published on the role of the microbiome, or the proposed role, perhaps we should say, in COVID-19, both in terms of how changes to the microbiome perhaps may make certain individuals more or less susceptible to COVID-19, and also the role that the SARS-CoV-2 virus has, or the impact that it has on the microbiome. And according to one paper, the proposal is that SARS could adversely affect the microbiome in a way that may actually contribute to long COVID, for the subset of patients that are developing long-term symptoms, even after the virus has been cleared. This one paper at least proposed the idea that one potential cause of that is the disruption of the microbiome that's caused by the virus itself. So it seems that there may be a bidirectional relationship here. And as someone who studies the microbiome pretty much on a daily basis, I'm sure that's not a surprise to you, given what was already known about the relationship between viral infections and the microbiome.
Yeah, I think it would be helpful to start a little bit with some background, which is, the gastrointestinal tract and the lungs are both body compartments that host microbiota or microorganisms. The lung is much smaller in terms of absolute count when compared to that of the gut. The gut's the largest reservoir of microbes in the body. And really the evidence is accumulating even before SARS-CoV-2 emergence that there's a bidirectional communication between the gut and the lung. And this is referred to and called the gut-lung axis. And so, this kind of crosstalk is involved in immune homeostasis and inflammatory cascades. And it's really believed that gastrointestinal inflammation can result in lung inflammation through this connection. And so the mechanism underlying this inflammatory shift from the gut to the lung is not completely clear yet. However, we think that dysbiosis of gut and lung microbiota is definitely one of the factors that play into this crosstalk. And so a lot of talk has gone back and forth, particularly in more academic circles, trying to pinpoint exactly, what is it? What is the nature of this cascade? Is it that SARS-CoV-2 infects the host, and then changes the gut microbiome, or is it that the gut microbiome increases the risk of severity when an individual gets SARS-CoV-2 and drives the severity of their outcomes? And I really want to focus on this one part, because this, what's called like the chicken or the egg, or at Seed we joke and call it the bacteria or the archaea, but it's really it's not clear. And it also reveals one of the biggest limitations of the most recent study that came out, which we can jump into right now, which is we don't have samples of these individuals prior to them getting SARS-CoV-2. And so That's kind of
Exactly.
the critical little bit of information that you want to say, well, what's really driving this outcome. And so if you look at this study, it was published in BMJ a couple of weeks ago, and it basically looked at stool samples from 27 hospitalized COVID patients, and the finding was that the bacteria in their guts seemed to influence the severity of the COVID cases. And this builds on two other studies that have come out over the past 12 months with a similar theme. One was in a clinical infectious diseases journal, and it found that the microbiome in people with COVID was different. And then another one in about 15 or so people that found that saw a lot of changes in the gut bacteria during hospitalization compared to non-COVID controls. And this is just the gut microbiota, not anything else. So the small sample size, the fact that we don't have information before they got the infection, and importantly for me, the fact that none of these studies really excluded people who'd previously been on antibiotics or any other types of conditions, which we know play a really big and have a long duration in the effect that they have on the diversity of your gut microbiome. And so I do want to say at least that the associations are there, and we know enough about the gut lung axis at large to say that it's certainly possible that gut microbiome drives severity of outcomes. I believe it does for reasons we can for very distinct reasons we can get into afterwards. But it's not that these new studies that have come out are somehow a smoking gun and that they've identified something beyond just the same types of studies that we've seen over the past year.
Yeah, it's reminds me a little bit about the of the vitamin D and COVID connection. Although there's an advantage with the vitamin D research because researchers have been able to find data sets where people did have a serum 25-OHD measurement done prior to getting COVID, oftentimes within the last 3 months. And then they can prospectively, study what happens to those people based on their vitamin D level prior to getting COVID, because there is some evidence that suggests that the SARS infection may actually suppress D levels. So that it's a similar question there of like, is it a chicken or egg or bacteria or archaea? But the advantage with the vitamin D research is that there are actually data sets where you can get that measurement prior to the patient becoming infected.
And then the other thing is, people latch on to community dynamics. In other words, which bacteria you can find because taxonomically it's very attractive. And all I mean by that is like you can do co-aggregated, you can you can map through Permanova and see the way the microbiome changes, and you can get species and strain level resolution. And it's from a data perspective, it's really attractive because you have so much data. And when you have that much hay, you basically can make your own needles, so it's very easy to find these types of taxonomic changes. But I want to give my same stump speech that I always give, which is the presence of a single or individual species is no indication of its function exclusively across a whole population. So different bacteria can do the same thing in different people. And so this kind of like species level obsession, I think, is more of a flaw of like what drives good and novel paper publication rather than I think what drives for a real deep understanding of what's happening. Now, that's not to say that there aren't biomarkers that we should use. And in the future, we're gonna look back, and even as soon as like 2 years and laugh that we looked at single species biomarkers as a way to understand what's happening in the gut, right? Like even in some of these more complex, like major depressive disorder studies, they're now starting to build more complex panels that include the presence or absence of a metabolite and the presence or absence of a bacteriophage. And then finally, when you stack on the presence or absence of one organism of a particular class, then you can start to get really, really high predictive power in things like cancer and depression. And, you start to get a lot more resolution. But the problem is very few data sets have that integrated level of data that you can start to build those more complex panels with. And so what I think is going to happen with SARS-CoV-2 is we're going to see that a combination of blood, serum data, cytokine, and gut metabolome, and maybe a little bit like gut species are gonna drive that, some predictive value. But it's not just going to be predictive for SARS-CoV-2. It'll probably also be predictive of severity of other viral infections.
Absolutely. And I mean, my take on this is this isn't, it's not just related to the publication of scientific papers. It's actually more of a philosophical bias that exists in the way that we approach health and disease, and study it. It turns up in nutrition a lot too. There's a food philosopher named Georgi Skrinis who coined the term nutritionism, which refers to the reductive understanding of nutrients as the key indicators of a healthy food. So this has been like our obsessive focus on individual nutrients, whether you're talking about macronutrients like saturated fat or cholesterol, or whether you're talking about micronutrients, like vitamins and minerals. And his argument has been that this ideology has really narrowed and in many cases distorted our appreciation of food quality, which is a much more meta concept, right? Like we study and pay attention to nutrients in isolation without considering the context that they're delivered in, right? Like is saturated fat going to have, if two people are following a diet with identical levels of saturated fat, but one person is consuming that saturated fat in the context of, French fries and donuts and lots of processed and refined food, and the other person's consuming that saturated fat in the context of, a healthy whole foods diet, I think most people would intuitively suspect that that's not going to have the same impact. But the way that science has been done on nutrition does assume that it will have the same impact. And I'm starting to see now trials that are being designed with this in mind. In fact, I know there's an RCT that's being designed now that's explicitly going to look at the role of saturated fat in a standard American diet versus a whole foods diet. So, I'm curious if you think that's playing a role too in how microbiome research has been done thus far.
And it's just like, I mean, it's so challenging to get good, clean samples that both in microbiome studies as well as diet studies, most often people rely on retrospective analysis. And so I think that really is what drives it. I mean, if you could recruit 100 people and get to that level of clustering and granularity and the type of saturated fat or the diet that corresponds to the presence of saturated fat, then I don't think that there's some inherent like desire to brush good research under the rug necessarily, but it's just so hard that usually what's available is you look at everything that you can test for, you look at everything you can analyze, and you try to see what patterns emerge. And sometimes the patterns emerge, with so many other factors being different, and then that starts to create a very skewed narrative. And so I don't think it's like it's malice per se, but it's definitely a huge limitation in the ability to study the role of diet and any complex, multi factorial system, whether it's a intake of food or whether it's the composition of the microbial composition within the gut, and you just see this in these complex communities. And so one thing that will really help is kind of like a focus on functionality and a obsession on mechanism. And I think that getting the ability to say, getting just a nature paper because you identified a species that's present or absent in some areas is that time's also, even in microbiome, kind of coming to an end.
Yeah. Yeah. I mean, I agree. It's multifactorial as it often is. It's usually not just one cause, but I think there are encouraging signs that we're shifting that model, and hopefully there'll be more funding available for those kinds of studies because, as you pointed out, they are more intensive to perform and require, a level of effort and investigation that's not always easy or possible. So.
But just really quickly to close the loop on SARS-CoV-2, I
Sure.
mean, one real example of how I think it drives it can drive bad policy making and drive bad medical outcomes is when, like let's just take everyone that follows SARS-CoV-2 has probably heard of this term cytokine storm by now. And it's this idea that partially driven through the gut microbiome, that the inflammatory cascade, or the way that your body responds by mounting an inflammatory response, is determined by the gut microbiome. And inflammatory and cytokine storm, or inflammatory cascade, is really just this like massive production of TNF alpha, IL-8, interferon gamma. I mean, the whole cluster of what you're looking for, that then causes scorched earth cells, and then a further cascade that causes a destruction of tissue and multi-organ system failure. But if you assume, like, if you go too quickly with and run with this, then a lot of a really nice article came out in JAMA that was titled something like, "Is cytokine storm in SARS-CoV-2 real?" And it's not that there isn't an elevation of cytokines, but when you look at in SARS-CoV-2 patients that have elevated cytokines and compare it to like the middle of the curve in people that have ARDS, which is the condition that SARS-CoV-2 leads to, that actually results in like 50 to 60% mortality. It's acute respiratory distress syndrome. It's still much lower. And in some instances, it's like 10% to 1% to 10% of the cytokine levels that you would see in median cases in previous viral infections resulting in acute respiratory distress. And so what that means is that, yes, cytokines are elevated compared to healthy controls, or even compared to people that recover quickly. But is it too early to say that we should be using drugs that block a healthy cytokine response in the treatment of it? Or is that just kicking the can down the road and increasing your risk of reinfection, or blocking the ability of the immune system to actually effectively induce an inflammatory response? And the immune system is such a delicate, and it's what makes immunology so challenging, is it's so delicate and balanced that when you move too far on one side, you pull from the other. And so it's very hard to say. But as a cautionary tale, I do want to say, like, those approaches that except in very severe cases might be too extreme. But what makes microbiota intervention attractive is it has this element of homeostasis, right? Where things like dendritic cells that play a very key role in immune homeostasis, by the way, in the healthy intestines, they present antigens, and then they have a dependent response regulating pro-inflammatory or anti-inflammatory phenotypes. And so it's that calibration, that like regulation that the microbiome has, which makes it attractive compared to other typical pharmacological interventions, where the stimulation can be responsive based on other factors that are occurring in the body. And so I think it's definitely too soon to say, well, is probiotic or prebiotic or dietary or targeting the gut microbiota modulation something which is going to, reduce the severity of SARS-CoV-2 infection. But, we know that the gut microbiota drives dendritic cells to prime these cells. We know that it stimulates IFN gamma production. We know that it can regulate pro-inflammatory Th1 cells. And with all of that information in mind, and especially with previous studies that show that respiratory tract infections can be mitigated through the type of, like, whether it's IgA secretion, or whether it's boosting Peyer's plaques or neutrophils or there's a number of different mechanisms that oral consumption of bacteria or modulating the gut microbiome can have very, very validated effects on regulating, kind of viral or respiratory infections. And so I know that the connection, the link is there, and people are going to be looking for a simple answer to this. But I think that just like the most I can say right now is consideration of the gut microbiome and the gut, kind of like the host microbe barrier is something which I wouldn't discount. I think is going to be, we're going to find that it does play a big role in, and again, not just this infection, but other viral respiratory infections.
Yeah, I mean, I appreciate your clarity there, and I'll reiterate that we don't have any direct evidence yet with probiotic interventions and COVID, but there are quite a few lines of evidence from prior research with other viruses, like, for example, bifidobacteria and lactobacilli studies have shown some beneficial effects against influenza. There's studies that have shown that probiotic consumption improves type 1 interferons, antigen presenting cells, natural killer cells, T and B cells in the lungs immune system. As you pointed out, probiotics have been shown to improve pro and anti-inflammatory cytokine balance, helping to clear viral infection from the lungs. We also have interesting research on the nasal microbiome, which is how you and I connected in the first place, right? Geeking out about that and Susan Lynch's work showing how reinoculation of the nasal microbiome can help reduce chronic sinusitis. So there are a lot of interesting lines of research. And for me, I'm just curious, and I think we should be exploring these because it seems to me that the benefits pretty far outweigh the consequences of this kind of exploration. Like you see lots of studies and stuff published in the media about, drugs and interventions that are being explored, for COVID, rightfully so. Some of those have potential significant downsides. And, they're also very expensive and maybe not even that accessible for a lot of people. And then you have an intervention here with probiotics that could be deployed on a pretty broad scale. As far as we know, there are not significant, that it seems less likely, and we don't know this for sure, but it seems less likely that there would be any adverse reactions or significant downside. But it's a little bit frustrating that often these kind of these nutritional interventions are typically seen as sort of second or third order and not worth exploring to the extent that some of the other interventions are being explored.
I don't blame, like mainstream science for blocking nutritional and secondary interventions, or at least having a very high evidence threshold before it's accepted. And it's just because there's so many bad actors that take advantage of I mean, you can't you really have to watch, like, yes, I would love to have a nuanced discussion about whether why, like, the AstraZeneca vaccine at a certain dose was only 60% effective, whereas at half a dose was 90% effective. But like, the problem is when you open up those gates, then it seeds the doubt for naysayers and conspiratorial theories to say, well, so you're saying that vaccines could be dangerous? And it's like, no, we're saying that the dosage is very important in driving, more work needs to be done in establishing dose response to efficacy, right? And so, like, I think that there's this, like, and we could go as philosophical as you want on this, which is, I think, what got us here in such a polarized place, which is, one side tries to kind of treat the other side, like withhold information that might allow for conspiracies to take hold. And in doing so, they actually seed and feed the very conspiratorial fire that drives the other side to believe that information is being withheld from them. And it's because people don't have attention spans, and people don't want to have a discussion and understand that the answer is oftentimes it could be both.
Yep.
And that's why these therapies aren't looked at, because the minute that, like there was a report in Nature Food Sciences that came out that was, well, two of them. One in American Society for Microbiology, titled "Does an Altered Gut Microbiota and Associated Leaky Gut Affect COVID-19 Severity?" And that is just a very, very interesting and deep scientific exploration of how intestinal permeability, which we know that diet and probiotics and other interventions can definitely affect. We know that can affect the severity of COVID-19 infection. And then another one that was in I think it was Nature, the Science of Food, the NPJ Science of Food, which is a Nature journal, which is a very nice review on the potential applications of probiotics and prebiotics for the prevention and treatment of COVID-19. And these are pretty balanced and high impact journals, and very clear and informative publications. But the minute that then becomes like a recommendation is the minute that you start to see companies taking ads out positioning their product as immune boosting. I mean, just walking down the street here, I see juice shops talking about the antiviral activity of their juices. And
Mm-hmm.
so I think that both sides need to but both sides are to blame. Actually, I don't I think one side is a little bit less specific, to put it kindly, than the other. But yeah, I mean, both people need to just get their head out of their ass and say, like, are we going to have a discussion? Are we going to get into this and have a discussion about it? Or do we have to continue, like, vacillating between these two extremes and shutting everything down that's in the middle because of fear that someone's going to, take it and run with it and commercialize something fraudulently? And I don't really have a good
Yeah.
answer to that.
Unfortunately, that polarized dynamic is not just occurring in one field of science and medicine. It's pretty much the nature of our reality at this moment in time, and that's a whole nother podcast. But let's talk a little bit about the gut heart axis, some research updates there, because that is relevant to COVID, and then we can kind of use that as a bridge to talk about some of the IBS work and other stuff that's exciting stuff that's happening in probiotic research.
Absolutely. So a new paper came out in Cell Host and Microbe. It was by a group at Mass General and Harvard. It was the senior author, I believe, was Ramnik Xavier. And this was a really interesting paper because, I mean, cholesterol's kind of like, just to start a little bit on like the story of cholesterol and what that really means. And a lot of your listeners are kind of like part of that like saturated fat camp that says like, well, it kind of matters, like dietary cholesterol, doesn't it matter where it comes from? What are the other features of it? Is it single-handedly an enemy, or is it just an actor? Is it the density of the cholesterol? I mean, there's so many other factors to this that, others before me have spoken about really well, like Peter Attia, for example. And cholesterol is kind of like rise from this obscure component in gallstone to like a main household word was really kind of like a defining story of the 20th century. And we first started with the fact that there were genes, and this is like a good like high level analysis of how microbiome and the human genome really play, right? Like we looked at genes in the host that were varied across the population, and that drove both the synthesis and reabsorption of cholesterol, right? And so cholesterol is synthesized by your body, and then it goes luminal, and then it's reabsorbed back up by your intestines and goes back into circulation. And so the biggest blockbuster drug in history works in modulating this human genome pathway, and that's statins, right? To keep cholesterol low. And so, but this doesn't, cholesterol lowering drugs are explain how pharmacology can lower cholesterol, but not really how pharmacology can lower cholesterol to reduce death. And it's that last part that's really important, because there's so much human genetic variability, and today it doesn't really adequately explain like these differences that are observed between people in complex processes like cholesterol metabolism. But we're starting to see that in the human gut microbiome. We can explain some of these clinical observations with a lot more sophistication. And so we know that the gut microbiome metabolizes drugs. We know it tunes the immune system. We know that it influences nutritional imbalances. It produces neurotransmitters. I mean, the connection between like heart disease and the human gut microbiota was a really early big success story for the role of the gut microbiome in these organ systems across the body. And to this point, cholesterol has not yet been directly linked to specific microbes or enzymes or functionality within the gut microbiome until this paper. And so what this paper really did was it shows that basically cholesterol, gut microbes convert cholesterol into a secondary or tertiary metabolite called caprostanol. And caprostanol, interestingly enough, is uniquely made by human gut microbes. In fact, people actually developed an assay to look for caprostanol, to see if there's contamination in the environment from like human sewer systems, because if you find it, it means that human gut organism metabolism is present wherever you're looking. And this step happens in soil a little bit differently than it happens in the human gut. And in the human gut, what happens is specific bacteria now convert cholesterol into caprostanol, and therefore they lower the total circulating cholesterol load, in consequence, the amount that can get oxidized, and we believe in consequence, the amount that can drive acute cardiovascular or cardiovascular or vascular outcomes associated with oxidized cholesterol and LDL and VLDL. And so what this paper really shows is a mechanism by which certain organisms convert that circulating luminal cholesterol that's just kind of floating through. Every time you eat food, it's released and it's floating through your digestive system, and then it's reabsorbed back up. But this is involved in the conversion of that. And I think that that's really important, because it's the use of modulating the microbiome in the future for people that have a genetic predisposition to produce high cholesterol or get high dietary cholesterol. These things can help kind of mitigate and keep those levels down because you're siphoning some of it off through the production of from cholesterol to coprostanol.
Yeah, that's fascinating. And of course, it's highly relevant because heart disease is still the number one killer in the US. And now we have COVID-19, which affects the heart, and anything that, makes the heart or the cardiovascular system more susceptible is something that we should definitely be paying attention to. I know there's also been some new interesting work, and we talked about the gut-brain axis before on the previous show. I've talked about it, probably at least 15 or 20 times on the podcast with various guests. It's one of my favorite topics, and I know that there's been some new interesting research there as well, particularly related to how the gut-brain axis may contribute to major depressive disorder.
Yeah, absolutely. So I kind of alluded to this earlier on when we were talking about SARS-CoV-2, but this was a study that was done in 200, it looked at over 200 people, and it kind of builds on the study, just to bring everyone up to speed. The study that we spoke about last time, at the time, had just come out in Nature Microbiology, and it was done with the Flemish cohort in Belgium. And Jeroen Reyes was the senior author on the paper. And in this paper, they found that first it was the observation that hundreds of neuroactive metabolites are made by the gut microbiome. And this isn't just kind of like your hallmark GABA production, but a whole cluster of never before really characterized neuroactive metabolites that have systemic effects and that cross the blood-brain barrier. And so what was found is that in this cohort of people that had major depressive disorder and were unmedicated, that two organisms that were responsible for driving a lot of this neuroactive metabolite production were consistently missing or were deficient in those patients compared to healthy controls or people that were not diagnosed with major depressive disorder. And I think it was Coprococcus and Dialister were the two organisms. And so this is really interesting because, it's actually like one question that we get so much from customers, from patients, from investors is around this idea of personalization, right? And in answering this question, I want to weave in a little bit of a refresher response to this kind of personalization story. And I'm sure everyone's kind of getting hit up left and right with like, a 2 or 3 part story, which is act 1 is everyone's microbiome is different. It's as unique to you as your fingerprint, which is true. Second is we have a test that can find out exactly how unique and different and special your microbiome is. Okay, awesome. With you so far. The sleight of hand is that third little act where they then come in and say, therefore, you need personalized probiotics or personalized dietary treatment or recommendations to fix these problems that are personalized and distinct to you. And that's the sleight of hand, right? It's the first 2 are fine, but it's that third part where you have to ask a little bit closer and say, okay, so what are you basing your intervention of? But what about my microbiome are you basing your intervention on? Would someone who does not have my microbiome also benefit from the same intervention? And have you tested this with a control to see if this is effective? And I haven't found a single group that can actually answer that question for more than 1 out of those 3, other than the one group in Israel that's doing it so responsibly, because they're looking at glucose metabolism, they're looking at insulin resistance. And so they have a very specific endpoint, right? It's not like this is you and your microbiome and this is what you need to be healthy. It's saying these are the foods that probably will spike your blood sugar more based on the organisms that you have in your gut. That, by the way, we validated in an 800-person trial and we published in Nature. And so like those are 2 very different studies, and that's insulin. But to bring this back now to the gut-brain axis, this study that came out recently in Science Advances is going to start to give us that level of visibility for major depressive disorder when it comes to gut-brain axis. And not on the intervention side, again, we're very far from that, but at least parts one and act one and act two, we're now going to get that visibility. And so what they did was they looked at all these people. They did the classic, people that were depressed had more Bacteroides and had less Blaudia and Eubacterium. They did like the species thing. That's where everyone else leaves it. Then they went and said that disturbance of amino acid metabolism from the metabolome is a hallmark in the gut ecosystem of major depressive disorder. And so now we have kind of this whole panel, right, of amino acid metabolism that leads us to say, when this is dysregulated, and it can be dysregulated in different ways in different people, but when there is a dysregulation of amino acid metabolism, there's a very high predictive value that there's going to be something that distinguishes people with major depressive disorder from healthy controls. And they put together a combinatorial marker panel that looks at the microbes, it looks at the metabolites, and it looks at a couple features and on the host side, biomarkers circulating in blood. And they developed something which can actually predict major depressive disorder in 80 to 90% of cases just by looking at this composite panel together. And I always say, if you want a diagnostic or a personalized company to be legitimate, or a technology to be legitimate, is can it be predictive, right? And so another good example of it being predictive is a scientist that we collaborate with a lot, Chris Mason. He's at the Cornell School of Medicine, Weill Cornell School of Medicine. He published a big paper on IBS and found a gut-derived biomarker for IBS that has that final test. It is predictive. If you have this biomarker or in this panel that they developed in a certain way, they can blindly send you 10 samples, 5 of them have IBS, and probably 4.8 of them they'll be able to predict someone has IBS just based off that sample. And until we get there, everyone else is just playing. And so this was really cool because it was nice to see that kind of evolution towards functionality, towards combinatorial biomarker panels that are put together, towards predictive values, like all the things that we really like to see in high quality science that separates it from kind of like the rest of that noise, which is just like another, large observational paper that like, it's really like looking for tea leaves, and then they come up with something and they publish it and then they never talk about it again.
Yeah, I'm encouraged by the quality of the research that's being done in these areas. And speaking of that, let's talk a little bit about some of the research that you have done and are doing. Let's start with the simulator of human intestinal microbiome ecosystem, or do you folks in the scientific community pronounce that SHIM or SHIME for short? Yeah. SHIME. Yeah, so tell us a little bit about what this is first, and then how you're employing it for your daily symbiotic and other probiotic interventions that you're considering.
Yeah, well, as you know, we developed a symbiotic formulation, which is a broad spectrum symbiotic. It has 24 distinct strains of bacteria across 12 species. And very much by design, the purpose here was to say, what are these features that you're really looking for, right? Or that are often dysregulated across a range of use cases that can have a rescue effect when you introduce a consortia like this into the gut, or when you consume or administer orally this type of a microbial consortia or cocktail. And so as part of that exploration, we're pretty incessant about interrogating the effect of it in very different systems, in human systems, and in simulator systems where there's limitations in human systems. You can get a little bit more immediacy, a little bit more granularity. You can actually look at it in response to different challenges. And then also like kind of genomically, right? Like mining the genomes of these organisms and others in our strain bank to say, well, what are these doing? Very similar to kind of like the enzyme, the genes that are responsible for encoding the enzyme in the cardiovascular, the cholesterol study we spoke about earlier. And so we're always interrogating this and other strains in our bank for these types of features. And some interesting new data just came back where we tested DS-01, which is this daily symbiotic consortia that the product that we've developed. And we wanted to answer really two questions, which is, the first is, everyone when they talk about probiotics talks about survivability. For the most part, you want the organisms to be alive and metabolically active to get the majority or to maximize the biological effect that these probiotic organisms can have. So you don't want dead bacteria. An entire industry has been spawned about soil organisms and spore forming organisms and acid resistant technologies that just kind of like try to speak to a lay consumer and say, well, just trust us, like this is survives really well. But then when you ask them for the data or for the publication, they don't have anything, right? Like they just rely on kind of generic features of the nature of spore formers to make that claim. And so what we wanted to do was we just wanted to like very effectively validate, you know, and why we wanted to validate it is because it took us 2 years to develop a 2 capsule system with varying density polymers so that you have a kind of a precision release of the bacteria in the upper small intestines, where most of the immune system is. And so you have the opportunity for these organisms to be biologically relevant in engagement with most of the immune cells before they just kind of like dump into the colon, right? And so this kind of took a lot of time, a lot of R&D, a lot of effort to develop this. And so we screened and compared this against, I don't want to get ourselves in trouble. And so I'll just say the number one, in air quotes, gastroenterologist or doctor recommended probiotic today.
Yeah.
It's available at all of your supermarket stores, and it's $100+ million franchise a year, and makes a lot of these same claims. And we started to test against that. And by the way, if you want to see exactly what it is, you can go look. We do disclose all of the products we screened against in our patent filing, and so there's you can that's a workaround to find out exactly all the products
Right.
that we tested it against. I mean, Chris, I can't explain how ridiculous it is that this product has 100% survivability, not 98%, but 100% survivability in triplicate that's being tested that we've developed. And when you compare that against this product, which has less than 10% survivability and less than 1 billion organisms that even begin to start with. And so I can say, based on this model with a lot of confidence, that there's very few, if any, organisms that are metabolically active when somebody buys this product. And like you just hate to see that, because like if you just said we don't know if this organism is surviving when a company sold their product, that would be one thing. But there's this whole industry which overstates survivability. Like voluntarily they say survivability is a feature, right? And it actually is a bug. And so we then tested this out against 16 of the next largest, both medical and commercial probiotics that are available. And these are ones that you typically find, you know, prescribed by practitioners. They're including like even like the very big medical probiotics, like against pouchitis, like VSL3. I mean, we went up and not a single probiotic in any of their current delivery systems or formats delivered absolutely more than 50% of the starting dose, not a single one. And so look, maybe something comes out that actually the organisms don't have to be alive and full in the future to do it. But like part of, I think what you see, this kind of like pissing contest of like how big your CFU count is it really doesn't matter, because bacteria die logarithmically, right? So if you start with 1 trillion, but you have a 2 log die off, you know, that's kind of the same as a 10 billion CFU dose that has 100%. And so it's not it's really important. And look, I don't want to overstate, like take that language, take that for what it is, because this assay that we tested was purely just survivability. So I don't want to overstate and say like therefore everything else is shit. But I can say with definition that the viability of active cells is not present in greater than 50% on any of the commercially or medically available probiotics that we tested. And we spent the money to test them because they wouldn't do it themselves, right? So we just said, like, let's just actually test everything head to head. And that part, at least, I mean, if anything else, I just want this to be a call to action for people to start publishing their data if it is conflicting with the data that what we've generated, because we just took products off the shelf, you know, and did it at every single stage and tested these things. And so what we can represent is that the delivery system that we have does actually deliver a precision small intestinal payload of 100% of the organisms, but by the middle of the small intestines in our product. And we don't see that in any other products that we've tested. And so that data was really interesting. And by the way, we tested a ton again, I'm not going to mention anything by name, but a very, very popular, several popular spore forming bacteria that are like extremely big in like the practitioner network. And that one actually had less than 5% survivability despite having spores. And so, maybe they go dormant for a very long time before they become active, but at least in this model system, there was not activity.
Yeah, and then I talked about this on our first show, and I've written and spoken about this a lot as a clinician. That this has all been very obvious to me just in using the product. So many patients that I've had either had no response to other probiotics or they had a negative response. And with seed, the daily symbiotic, it's just number one, been so much more effective, and number two, so much better tolerated. So it's not surprising to hear that, but it's awesome that you have done that research. Let's definitely cover the IND status for the daily symbiotic for IBS, and then let's talk a little bit about pediatric and what your plans are there. And that, because I'm, as you know, super interested in that. And just, this is maybe a good spot for full disclosure here. I mentioned this last time, but I am an advisory board member of SEED because I believe so much in what they're doing and how they're approaching the creation of these interventions from, with a very solid evidence base, as I'm sure you've gathered by listening to this show. But yeah, tell us a little bit more about IND for IBS and pediatric
Yeah, well,
plan.
and just to blaze through these last parts, then the Scheim system was also tested on the daily symbiotic after both ethanol, so alcohol consumption. We used vodka in the experiment, as well as a high dose of antibiotics, and that paper is going to be submitted next week. And so maybe in the next few months, depending on reviewers, you saw a very nice recovery shift, a butyrate rescue that was higher than even the individuals before taking antibiotics, their naive microbiota when it was transplanted into the system. And so that's something which, now that the paper's finished now for submission, we can speak about. So regarding IBS, where to start on this one? I mean, the FDA asks you to file an IND application, which is called an investigational new drug application, which is basically for anything to be FDA approved, you have to file that and then go through phase trials under the oversight of the agency. And so they ask for this if you ever want to test your product, and even if it's not intended to make prevention or treatment of disease claims, if you want to test this in a patient population that has a defined condition, and if you want to assess recovery outcomes. And so that was like almost a year process where we're putting paperwork together back and forth and communicating with them. This trial is being done at the Beth Israel Teaching Hospital at Harvard Medical School, and in a patient population with IBS type C and type M. And we announced a couple months ago that we got that authorization, the acceptance of that authorization, and are halfway through recruiting for this trial. So a small plug: if you have IBS and you're in the greater Boston or Cambridge area and you'd love to participate in this trial, we're still recruiting, and please reach out to the team at Beth Israel Hospital, and we'd love to have you in this trial. And we're just really excited, partially because how compelling. I mean, the thing with IBS is that there's such a psychosomatic component to it, and I don't mean that in like a way saying that it's a contrived, but just that there is such a bidirectional relationship between stress, the gut-brain axis, and all these other factors, and the severity of these conditions that are also broadly defined, like IBS, and response rates. And particularly when people are really excited about a new intervention, then they can feel a temporary improvement. And so the problem with IBS trials is just that the placebo responder rate is so high, and some instances it's over 40% or 50%, which is like higher than what you'd find in many pharmacological interventions for IBS or for IBD. And so it's made studying these really challenging. And so this is a really nice trial where we kind of hope to beat that placebo responder rate, and with an efficacy rate that's even a lot higher, and have some screening techniques that allow us to get much cleaner data. And so that trial is underway, and IBS affects a lot of people in the United States, and even if it's not for resolution of the disease, but small things like symptomatic improvements in quality of life that we hope to obtain from this. At the very least, we just hope to say, like, this is when things like the when reports like those from the American Gastroenterological Association come out and say that you need to test your products in a patient population to make sure that it's well tolerated before even recommending them as nutritional inputs, we just hope to follow suit in those types of calls to action and engage with the regulatory bodies instead of hide from them, and hope to set an example and encourage other companies to do the same, which is if you want to make claims on IBS, which you should never do it as another probiotic company, or you have to go through the process, and you have to study it, and you have to have data to be able to provide it to physicians and practitioners and clinicians to say that this was well tolerated and the microbiota remains stable, and maybe if there are improvements in symptoms, then you can report those as secondary endpoints, but at least to just back up that this is going to be something that's safe to go into that targeted population. So that's what's going on with IBS, and so we hope that we'll get that publication out this year, and kind of a sneak preview on our next big launch. And right now, it's kind of just open to insiders and current users only. Is our pediatric product, and this was very much driven by the same methodology that we had in the development of DS-01, which is the cultivation of a strain bank and the development of strains that had really, really strong mechanistic and clinical data that drove them, and improvements in the delivery technology and the ability to deliver that payload where it needs to go, with the use of delivery technologies or microencapsulation systems, which is what we're using in this product. And lastly is just like a very clear evidence base that is applicable across a wide range of indications in a pediatric population. And so here we're looking already at established strain-specific, pediatric-specific outcomes in the gut-skin axis, and particularly, dermatitis and eczema-like symptoms in children, and this just patchiness and the modulation of the gut-skin axis in that way. As well as a new study, which is actually under review and hopefully will be published soon in Frontiers in Pediatrics, which were two strains that are in our strain bank in this product that were in a 400-person plus trial looking at airway allergy and asthmatic response and severity of asthmatic response in a pediatric population, and a significant reduction in events, right? Asthmatic events in the intervention group, the group that got the probiotic, compared to a control which just got the placebo. And so this is everything you want to see. It's a very large scale trial. It's an interventional study. It's randomized and controlled. It's multi-centered in site, and it's going out for peer review, and it's going to be published prior to the launch of this product. And so really kind of starting to piece together these other areas, right? Or these very interesting use cases of probiotics in a specific and defined population. And the last is that we have our own trial on the formulation that's being a new trial, I should say, on constipation in children, and modulation of the gut microbiome in children, particularly in children that have either long bathroom, like a long time in the bathroom, slow intestinal transit time, less than 4 bowel movements per week, poor stool consistency, and a whole panel of like digestive and gastrointestinal mediated outcomes in children. And so far that trial is going really well too. So very, very exciting, I think, to bring that same level of rigor to a pediatric population with pediatric specific studies. And I think it's really important, particularly when parents look to make decisions for their children. I think that having that evidence base and having that established beforehand is something which is really important to parents. And so I hope it's very effective and it's able to be helpful for a lot of parents and their children.
I agree 100%. Raja, thanks so much for joining us again. If you'd like to try seed, you can go to kresser.co/seed to check it out. And Raja, just kudos for all the phenomenal work you all are doing. It's really exciting to see the rigor with which you're approaching the research, and refreshing as a clinician who's been in this space for some time and seen so many of the claims that are made that are really just unsubstantiated. So it's really refreshing to have a company that we can really depend on that's doing this really rigorous research to ensure the quality and reliability of their products. So, and always fun to just geek out with you and talk about the science. And I know we have a lot of listeners that appreciate that as well. So we'll definitely have you back on again in the future to talk about what's new. And thanks again for joining us.
Always a pleasure. Thanks so much, Chris.
All right, everybody. Thanks for listening. Keep sending your questions to chriskresser.com/podcast question. Take care, everybody. That's the end of this episode of Revolution Health Radio. If you appreciate the show and want to help me create a healthier and happier world, please head over to iTunes and leave us a review. They really do make a difference. If you'd like to ask a question for me to answer on a future episode, you can do that at chriskresser.com/podcast question. You can also leave a suggestion for someone you'd like me to interview there. If you're on social media, you can follow me at twitter.com/chriskresser or facebook.com/chriskresserlac. I post a lot of articles and research that I do throughout the week there that never makes it to the blog or podcast, so it's a great way to stay abreast of the latest developments. Thanks so much for listening. Talk to you next time.